Now that we know the structure of the biofilms, let us go on to find out how biofilms are actually formed, or what are the various steps that lead to the formation of a biofilm. There are 5 main phases in biofilm formation. The first phase is a reversible attachment phase, where the microorganisms just adhere on to the surface on which they are going to form the biofilm. The next step is an irreversible attachment phase, where there is a change in the phenotypic expression of the microorganisms such that they bind more effectively onto the surface. The third step is the step where the biofilm actually gets formed, where proliferation and production of the extracellular polymeric substance is the main reason for the biofilm structure to be formed. The fourth phase is the phase where the biofilm matures and the various water channels are formed and the biofilm thrives effectively. The last phase is the phase where dispersal or detachment happens. This is a phase when microorganisms are dispersed out from the biofilm and get the capability to re-establish themselves on newer surfaces. So, let us now start looking at every step in a little more details. The first step, as I said, is the reversible attachment phase. In the reversible attachment phase, the cells attach to the surface, which we call as a conditioned surface, weakly with the help of certain adhesion molecules. Any surface that is exposed to a liquid environment immediately gets conditioned with organic molecules that are present in that aquatic environment. Say for example, in natural environments, the polymers that get coated on the surface are the normal polysaccharides or proteins that are present in the vicinity. Whereas in setups like, for example, the human body, the surfaces can get conditioned with substances like saliva, as in case of the dental biofilms, or with the lymph or the nourishing fluids like blood on tissues or on bones. So, these substances normally condition the surface and aid in attachment of the cells in the biofilm. The attachment of the cells in the reversible phase is by weak forces, which are forces like the non-specific forces, electrostatic forces, van der Waals forces, and the forces which are not going to bind the bacteria very firmly onto the surface. Attachment of the bacteria to the surface also depends on the roughness of the surface. It also depends on the charge of the surface, and it also depends on the substances that have conditioned the surface. Attachment to the surface also depends on the hydrophobicity of the surface. It has been observed that biofilms are better formed on hydrophobic surfaces like Teflon rather than those formed on surfaces like metals or glass. The next step is the irreversible attachment. Here we see that the bacterial adhesins play a major role. Like for example, the type 4 pili or the flagella of the bacteria help in creating an irreversible attachment onto the surface. Once the microorganisms adhere to the surface irreversibly, they multiply, and the multiplication of the microorganisms attached to the surface leads to a phenomena which is a very interesting phenomena of quorum sensing, that is cell to cell communication. Quorum sensing between the microorganisms leads to expression of different genes in the microorganism and therefore a different phenotypic expression. Quorum sensing can lead to upregulation and downregulation of different genes in the population, thus converting the population from a planktonic population to a sessile population. Quorum sensing also leads to the expression of the genes for production of extrapolymeric substances, and thus now the population has adhered more effectively onto the surface. The extrapolymeric substance that is formed by the microorganisms in the biofilm in this step of biofilm formation not only allows the microorganisms to adhere onto one another, but also allows the microorganisms to adhere firmly onto the surface, thus making the biofilm more stronger. The interactions that can happen with the surface can be interactions of the hydrophobic type or the ion bridging type. In this step of biofilm formation, the EPS that is formed is a very important component. This EPS helps the biofilm to adhere, to get a 3D matrix. It also helps in retention of water, in nutrient circulation, and certain important aspects like a collaborative behavior, especially when we talk about biofilms formed when symbiotic relationships happen on plant root surfaces. Biofilm EPS is also responsible for a very important aspect of genetic exchange, when the sheer proximity of the cells allows certain genetic elements to be transferred from one cell to the other. The microorganisms in this biofilm EPS matrix also remain protected from the external environmental stresses and predation. Moving on to the next step of biofilm formation, that is the maturation of the biofilm. The biofilm that had initially formed during the proliferation phase further now expands with more and more amount of EPS formed, and then forms the typical 3D matrix with water-filled channels, and later on into a very well scaffolded structure like the mushroom shape structures. In the phase of maturation, one also sees a complete change in the pattern of gene expression, also certain phenomena like loss of certain appendages like the flagella. The 3D matrix gives mechanical stability to the biofilm and also lays down a boundary for the formation of the heterogeneous structure in the biofilm. The next and the last step in biofilm formation is the dispersion of the biofilm. Environmental stresses like antimicrobial stress, physical stress like change in the shear, or environmental issues like change in temperature, pH, can lead to the dispersion of the biofilm in the form of slowing off of the extrapolymeric substance. Sometimes this can also happen because of the presence of enzymes that can degrade the extrapolymeric substance. Dispersion of biofilm also happens because of a change in the gene pattern expression. It is observed that genes for flagellar synthesis and EPS degradation get upregulated during the phase of dispersion, whereas genes for EPS formation, for adhesins, etc., get downregulated. Dispersal of biofilms can also happen because of environmental stress like change in pH, change in temperature, or change in nutrient availability. It has also been observed that autolysis can initiate biofilm dispersion. So, here is a picture that is summarizing the steps of biofilm formation. The first step of substratum preconditioning by ambient molecules, as I had earlier said, is required for the reversible attachment of the microorganisms. one can see that the cells are depositing and also they are adsorbing and desorbing from the surface, indicating that these steps are reversible. Once the adsorption happens irreversibly, the cell-to-cell signaling and the onset of exopolymer production initiates. This leads to the formation of a mature biofilm with more and more nutrients seeping through the biofilm, oxygen gradients getting formed, and replication as well as polysaccharide production, giving rise to a proper mature polysaccharide based matrix in the biofilm. The last step is the detachment and erosion and slowing off of the biofilm that can lead to dispersion of the microorganisms. Here is another diagram that is helping us to understand the steps of biofilm formation in correlation with the changed gene expression patterns in planktonic and sessile populations. It has been observed that certain genes get upregulated and certain genes get downregulated during the biofilm formation. Upregulation usually happens of iron sulfur metabolism, lipid metabolism, amino acid and carbohydrate transport, biosynthesis of secondary metabolites, stress responses, and genes encoding efflux system components. Whereas downregulation of DNA repair genes in the biofilms has been observed. This is a very interesting phenomena because DNA repair genes in the biofilms that get downregulated can decrease the repair mechanisms and therefore increase the frequency of spontaneous mutations. This leads to novel genetic traits that are formed amongst the biofilm bacteria.